Hello students, good evening.
Good evening, sir.
So we'll start the session. I'll share my presentation. Share my screen. So please let me know if my screen is visible.
Yes, sir, it's visible.
Okay. So we'll start the session. So let me introduce myself first. Myself Ankit Kumar Malik, a PhD scholar as well as PMRF fellow from IIT BHU, and the course instructor for the same is Professor Sanjeeva Srivastava from IIT Bombay. So the topics for the day are DNA tools and biotechnology third, DNA tools and biology fourth, fifth, sixth, and clinician perspective third. So we'll start with the question one of last year assignment. So question one is, so which of the following statements are incorrect about Sanger sequencing? So the options are, hello, am I audible to all?
Yes, sir, audible.
Okay. So the statement A is dideoxynucleoside triphosphate growing terminates growing DNA strand because it lacks three ending OH group. The next statement is synthesis of new strand starts at 5 end. And statement C is fluorescent detector senses the color of each fluorescent tag. And DNA template primer polymerase and deoxynucleoside triphosphate and dideoxynucleoside triphosphate are required as the reagents for performing Sanger sequencing. So, anyone would like to answer this question? Mm-hmm. Anyone? So, anyone know what a Sanger sequencing is?
Sir, Sanger sequencing, the DNA is mm, uh, cut by using chain termination method.
Mm-hmm. Correct, correct.
Um,
And what is the principle behind this Sanger sequencing? Principle?
sir, principal, sir, I don't know. Mostly for this question, I'm feeling A option is incorrect actually.
Okay. And what about option B?
Um,
Okay,
Option
correct.
B is synthesis. Actually, like the orientation will be from 5 dash to 3 dash actually of the template strand. So I think option B may be right.
And option B is incorrect. It do grow
Acha, okay,
DNA
sir.
strand because it lacks three end OH groups. So here the option two, statement two is the incorrect statement. So let me go deep and detailed information about Sanger sequencing. So here this Sanger sequencing. So Sanger's method of gene sequencing is also known as dideoxy chain termination method. So it generates a nested set of labeled fragments from template strand of DNA to be sequenced by replicating that template strand and interrupting the replication process at one of the four bases. So there are four different reaction mixture are produced that terminates, that is A, T, G or C respectively. Then I've asked the principle behind the Sanger sequencing. So it works on the principle that a DNA primer is attached by hybridization to the template strand and deoxynucleosides triphosphate, that is D and TPPS, are sequentially added to the primer strand by DNA polymerase. Also, the primer is designed for known sequences at third end of the template strand. So that's why this statement A is the correct statement, and statement B
Okay,
is
sir.
and following that So this is the principle. So here the two things are there. Then deoxynucleoside triphosphate and dideoxynucleoside triphosphates. So here the reaction mixture contains dideoxytriphosphates, nucleoside triphosphates along with the usual DNTPS. So if during the application dideoxy nucleoside triphosphates is incorporated instead of usual deoxy nucleoside triphosphates in the growing DNA strand, then the replication stops at the nucleotide strands. So based on these principles and the information which we have, the statement B is the incorrect statement. Rest other are the correct one. Moving further to the next question, it's two. So it states that a food technologist wants to compare the gene expression difference of yeast of Kashmir and Kanyakumari. So how should he design the experiment using microarrays? So whether he can extract total RNA from the samples and label the hybridized on the slide, or other he can extract the total DNA from the samples and label the hybridized on the slide. Also, he can spot cDNA on the array, extract total RNA from the samples, and label and hybridize on the slide. And the last, what he can do is he can spot either of DNA or RNA on the slide. So, what are your suggestions on this question? Anyone would like to highlight this?
So mostly he can extract the RNA and he can hybridize it on the slide. Like DNA codes for the RNA. So
Okay.
mostly like for the gene expression, like RNA um, is the most like you can get the information easily from RNA. So
Okay.
option A.
Option what?
Sir, option first one A.
Okay. So, do you guys have attended the yesterday's session also?
So yesterday didn't see any session actually, like in the announcements part.
Okay. Actually, this course has two sessions. One is on Monday 7 to 9 and another one is Tuesday 7 to 9. So we both TAs are covering same assignments. So I thought that you have attended there also because this question and answer all are covered in the yesterday session also. So anyone of you guys have attended the yesterday session? Anyone?
No, sir.
Okay, so you guys are the freshers on my sessions. I thought some of the
Yes,
students
sir.
are attending both the sessions. Okay, that's cool. So, what you are saying, option A, na?
Yes,
He
sir.
can extract total RNA from the samples and label and hybridize on the slide. Okay, let's see in the uh, concepts and basics principle. Then we'll get to know either A, B, or C or D. So, as per my opinion, the option C is the correct one. So, he can spot cDNA on the
Okay,
yeast,
sir.
extract total RNA from the samples, label and hybridize on the slide. Now, I'll explain why the option C is the right one. So, first we'll look what microarrays are. So, in this question, so a food technologists want to compare the gene expression. So, designing an experiment to compare gene expression differences between yeast strains from Kashmir and Kanyakumari using microarrays, which involves careful planning and executions. So here, microarrays are powerful tool that allows researcher to analyze the expression levels of thousands of genes simultaneously. So here we'll go through step-by-step guide on how the food technologist can design the experiment. So here are some steps are given here. So we'll go one by one. So the first step is he'll define objectives and hypotheses about the particular gene expression. So as clearly stated, the goals of the experiment, for example, so the food technologist might want to understand how environmental factors influence gene expression in yeast or from different geographical locations. So further, he'll formulate specific hypotheses related to the expected gene expression differences. Then he'll go and select the yeast strains. So choose a representative yeast strains from both Kashmir and Kanyakumari, which ensures that the strains are well characterized and have known genetic backgrounds. So further, the strains should be cultured under identical conditions to minimize environmental variations. And the third step is sample collection. So here he'll collect the yeast sample from both the locations, from Kashmir and Kanyakumari. And further, the samples should be taken under similar growth conditions to minimize any confounding factors. So further, he'll harvest cells at a specific growth phase to ensure uniformity. And the next step is RNA extraction. So in RNA extraction, he'll extract the RNA from the yeast samples. RNA represents the active genes and serves as the basic of microarray analysis. So by using a reliable RNA extraction method to obtain a high quality RNA. Further, that food technologies will synthesize cDNA and label it. So here he'll convert the extracted RNA into complementary DNA, which is cDNA, and label it with some fluorescent dyes. So typically different dyes are used for two samples being compared, such as Psi3 and Psi5. So here also you can see the Psi3 and Psi5 were used here. Then so these labeling allows for the discrimination of gene expression levels between two samples. So here as you can say the sample 1 is stained with the Psi3 and sample 2 is stained with the Psi5. Then further the next step is microarray hybridization. So here he'll hybridize the label CDN samples to a microarray slide which contains containing probes for thousands of yeast genes. Then the next step is find image acquisition. So using a microarray scanner to capture images of the hybridized microarray, and the data analysis will be done. So in next step, he'll perform some statistical analysis on the microarray data to identify genes that show how significant differences in expression between the yeast strains from Kashmir and Kanyakumari. Then the validation and interpretation and conclusion will be done by the food technologist. So at last he'll interpret the results in the context of experiments objective, whatever he has objectified and hypothesized in that. So according to this, all these steps. So here, wait, huh. So the options will go through. He'll not deal with any total RNA. It's complementary DNA. I've told na here. So he'll deal
So,
with the complementary DNA according to these principles. So he can support the complementary DNA on the array. Then he'll extract total RNA from the samples and label it and hybridize on the slides. So according to these theories, so option C is the correct one. So we'll move further to the next question. So the third question states that Nikhil works in a pharmaceutical company and wants to launch a new therapeutic protein for which he intends to manufacture that protein on a large scale. So he thought to purify the protein using affinity chromatography. So he asked the technician using N-terminal and his tag for better protein purification. So what is the important factor to consider while choosing the correct host for expression of the recombinant protein? Is it length of the gene, or the gaps between the exons and introns, or the presence of transposons, or at last post-translational modification? So anyone would like to answer this with some strong proofs?
so gaps between exons and introns actually, because exons are coding part of the DNA, introns are non-coding part. So if you remove the non-coding part of the DNA,
Okay,
then the coding part of the DNA will form the protein actually.
okay. And what do you think about post-translational modifications? Just a question. What do you think? What is post-translational modification? Just a hint. Mhm.
Sir, after
So, you know what is
sir, from mRNA to cDNA,
okay?
that is translation actually. So the modifications that happen from mRNA to cDNA,
Mhm.
so those are post translational modifications.
Okay. So, in that modifications, this N-terminal and histex protein purification has been done. So, I'll show you. I'll explain you These principles. So according to me, the most important factor he should have considered is the post-translational modifications instead of the exons and introns.
Okay, sir.
I'll explain. I'll give some strong proofs. So the question states Nikhil works. So when he chooses the host for the expression of a recombinant protein, so several factors needs to be considered to ensure successful and efficient protein production. So he must be use of an N-terminal histag for protein purification using affinity chromatography is a common and effective strategy. So here are some key factors that Nikhil should consider while selecting the host for expressing his therapeutic protein. So these are some factors which he should have considered. So we'll go one by one. So we'll start from the compatibility with the expression system. So the first, he'll choose an expression system that is compatible with the cloning expression vectors, which were available in the pharmaceutical company. And the common expression systems include some bacteria, yeast, or any mammalian cells. And the next step is post-translational modification. So in this modification, in this phase, so that N-terminal his tag protein purification has been done. So let me clear what PTM is. PTM stands for post-translational modification. So post-translational modification plays a crucial role in the expression and functional maturation of recombinant proteins. So after the ribosomal synthesis of a protein, so PTMs involve the covalent alterations of amino acid residues and can significantly impact the protein's stability, localization, and activity with some other molecules. So some common post-translational modification includes phosphorylation, glycosylations, acetylation, methylation, and disulfide bond formations. So these modifications contribute to the folding and structural integrity of the protein, influencing the biological activity and half-life. Also, in the context of recombinant proteins, the choice of expression system becomes paramount as different hosts which have varying capabilities to perform specific post-translational modifications. So here he can technician he should ask technician for the N-terminal histag for better protein purifications. And in the next step, protein folding and aggregations. In the third one, so he should have choose some different hosts with varying capabilities for proper protein foldings and minimizing aggregations. And after that, he should have checked the expression level and yield. So after that, he will evaluate the expression levels and yield of the recombinant protein in the chosen host. So while high expressions are desirable for efficient production and certain hosts may be better suited for expressing specific types of protein. And the next step which we'll go through is cell culture conditions. So in cell culture condition and stability, he should have considered the ease of handling and scalability of cell cultures for large scale protein productions and evaluate the stability of the chosen host cell line for consistent and reproducible protein expression over multiple generations. And after that cell line stability, he should have moved further for purification efficiency. So he has chosen the purification efficiency by using the post-translational modifications, which in which he should have he should be have compatible with the chosen host. Verify that the chosen host system can effectively produce the tagged protein, and that the tag does not interfere with some other protein functions. And the last, he will go through some cost and resources and some downstream processing. According to all these theories and some principles and some facts, we'll, we can say that Nikhil should have chosen the post-translation modification as an important factor to consider while choosing the correct host for expression of the recombinant protein here. So according to me, the option D is the correct one,
Yes, sir.
which is
Got it.
post-translation modification. So we'll go to the next question. So in next question, it states which of the following enzymes is used to synthesize complementary DNA library but do not synthesize a genomic library. So the options are restriction enzymes, DNA ligase, reverse transcriptase, or DNA polymerase. So anyone? Okay, let me ask someone. Or let me ask someone. This is an easy question, by the way. Let me. Anshi Agrawal. Anshi
Yes,
Agrawal,
sir.
are you on the call? So
Yes,
what
sir.
what is your opinion about this question? What do you want to say?
Sir, I guess reverse transcriptase.
Okay, and the reason behind reverse transcriptase?
Uh,
Mm-hmm. The, the answer is correct. I'm asking the reason why did you say reverse transcriptase? The answer is correct. It is reverse transcriptase.
yeah, because like opposite ऐसे करते तो मतलब reverse transcriptase होता है.
Sorry. Mm-hmm.
Like
Go
I
ahead. Go
can't
ahead.
explain properly, but I know the
Okay.
answer
Okay.
is reverse
The
transcriptase.
answer is correct. It's a reverse transcriptase. So anyone else would like to highlight this? Why reverse transcriptase? Anyone?
Sir, actually, reverse transcriptase is used for um, reverse central dogma. Actually, when yeah,
Exactly. Okay. So let
reverse
me.
central from mRNA to cDNA.
Okay.
So what happens is, like the other enzymes, like restriction enzymes, DNA ligase or DNA polymerase, they are used in some or the other function, like DNA polymerase in polymerase chain reaction, and
Mm-hmm.
restriction enzyme to cut the gene fragments, and DNA ligases to join the plasmid, join the gene of interest to the plasmid, so that it can introduce. So they are they are useful in genomic library in some or the other way. Reverse transcriptase is not that useful.
Okay. 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 Siddharth. Your name is Siddharth, right?
Yes, sir.
Okay. So the reverse transcript is the right answer. So let me go through some principles and theoretical portion behind this question. So first we'll know about what is cDNA library is and what is and how it is formed and what is reverse transcriptase. So first we'll go what cDNA is. So cDNA library is a combination of cloned complementary DNA in which fragments inserted into collections of host cells, which constitute some portion of the transcriptome of the organisms and stored as a library. And complementary DNA is produced from fully transcribed mRNA found in the nucleus and therefore contains only the expressed genes of an organism. So similarly, tissue-specific complementary DNA libraries can be produced. So coming to the question, Hmm. So coming to the question, when constructing complementary DNA library, so the key enzymes used is reverse transcriptase, which is a critical enzyme that plays a central role in the conversion of RNA into complementary DNA. So this process is known as a reverse transcription. So in a complementary DNA library, the goal is to capture the expressed genes within a particular cells or a tissue at a specific point in time. So since mRNA, that is messenger RNA, serves as a direct transcript of the actively expressed genes, reverse transcriptase is utilized to synthesize the corresponding complementary DNA from the messenger RNA templates. So okay, so am I clear?
Yes, sir. Clear.
What reverse transcriptase? Why reverse
Yes, sir.
transcriptase? So Anshi, are you clear? Am I clear?
Yes, sir.
Okay, so moving further to the next question. So the question 5. So here we have some assertion and reasons. So below there are two statements labeled as assertion and reason regarding the production of golden rice. So choose the correct options. So the assertion says, so eating golden rice will prevent vitamin A deficiencies that cause blindness and immune system problems. And reason behind it is that it produces beta carotene, which is a precursor of vitamin A. So here we have some options. So option A is both assertion and reasons are true, and reason is the correct explanation of the assertion. And option B states that the both assertion and reasons are true, but reason is not the correct explanation of the assertion. And option C states that assertion is true, but reason is false. And the last option we have is both assertion and reasons are false. So anyone?
Option A,
Okay, and why option A?
because golden rice is actually rich in beta carotene, and beta carotene is a precursor of vitamin A.
Exactly, Sanjay. The option A is the correct one. So it says that both assertion and reason are true, and reason is the correct explanation of the assertion. So let me go through some theoretical portion of the same. So here we have uh, some wild type rice, golden rice one, and golden rice two. So let me start from the basic things. What golden rice is? So golden rice was developed to address vitamin A deficiencies by incorporating genes from other organisms such as bacteria and daffodils are there, so into the rice plants. So these genes enable the synthesis of beta carotene in the rice grains, and beta carotene is a pigment that the human body can convert into vitamin A. And the next slide. So rice is a good source of vitamin B, that is thiamine and niacin, but poor in vitamin A normal rice. So we have some urgency to produce to translate the normal wild type rice into some golden rice. And after converting, why the reason? Because normal rice is a good source of vitamin B, but it lacks in vitamin A. So that was the reason we have produced golden rice, which is a transgenic line of japonica rice variety called type P309, which was developed by Ingo Procharitis and Peter Bayer, utilizing some transgenic Japanese technology. And so here golden rice is a genetically modified and contains beta carotene in rice grain, and carotenoids are the precursors of vitamin A. And golden rice refers to the golden color of rice grain due to its provitamin A. And golden rice was developed by using two genes. Here, as you can see, it was developed by using two genes called phytine synthase and lycopene beta cyclase from this daffodils and ervina, which it is in Eurydogra. Sanjay. So the option A is the correct one because it produces beta carotene, which is a precursor of vitamin A. So here the option A is the correct one. Okay, so we'll move
Yes,
to the
sir.
next
Got it.
question. That is question six. Question six states that transgenic plant cassava is said to have the ability to fight world hunger. So this is so as it has been made to produce iron and beta carotene. Which of the following can be used as a technique to insert the recombinant plasmids with the iron and beta carotene gene producing genes into the cassava plant cells? The options we have here's are electroporation, electrophoresis, polymerase chain reaction, and chromatography. Let me ask from someone. Wait. Uh, uh, uh. Shrimant Mune, are you on the call?
Yes, sir.
So
I'm on the call.
Shrimant Mune, so what is your opinion about this question? So which
One second, sir.
technique? Hello,
Yes, sir. Yes, sir.
am I audible to you?
So, yes, sir. Am I audible to you?
So what is your opinion about this question?
So, it's a RDNA technique, right?
Huh.
So, transgenic plant cassava is right.
I think you are.
Uh,
Uh-huh.
polymerase, polymerase chain reaction, but no, it's for DNA.
And why
They're
polymerase chain
asking for
reaction? You have
DNA.
some strong evidence for this.
Uh, it's like the means what I'm thinking, sir. If I'm right, you can just
Mm-hmm.
correct me. If I'm wrong, basically. So this iron and beta carotene we can do in RDN technology, that is plasmid.
Okay.
What we are in biotechnology, and after
Mm-hmm.
that, after inserting that DNA segment, as it's a plasmid, so we may not have that much of the DNA. So we can go with that another technique, that is PCR. So in that, denaturation annealing, and then we can get the whole lot of DNA.
Okay.
But I don't think it's actually the right way of thinking. That. But electrophoresis provider, nothing. Out of all that four option, I think C is the best option
So this
according to
PCR
my knowledge.
polymerase chain reaction may have some disadvantages also. So anyone
Okay.
else would like to answer this question? Then we'll.
Electroporation A.
Okay. And who said electroporation? Vanshika Garg.
Yes, sir.
So any strong reason behind this electroporation?
Yes, sir. Actually, electroporation is a kind of a technique in which we use electric pulse to
Okay.
create holes in the cell membrane, and
Okay.
it allows the foreign gene or protein to actually enter the cell. So, if we want to insert the recombinant plasmids with
Mm-hmm.
iron and beta carotene gene producing, then we can use this technique.
Okay. Exactly. The option A is the correct one. Electroporation is the best technique to insert the recombinant plasmid with the iron and beta carotene genes. You are correct. The option A is the correct one. Electroporation. Okay. So my screen is visible now to all.
Yes, sir. Visible.
Yes, sir.
So we'll go that why electroporation. So before electroporation, we'll go some theoretical portion of the term called before understanding the electroporation thing. We have to understand agrobacterium mediated transformation. So the technique commonly used to insert recombinant plasmids. If one more thing in these options, if we have if the question the professor should have included the agrobacterium mediated transformation. So we should have selected electroporation and agrobacterium mediated transformation. Both were the correct answers. As he has not included the agrobacterium mediated transformation, we have to select the electroporation technique only. So here we'll go with the electroporation things. So agrobacterium mediated transformation is a widely utilized genetic engineering technique for introducing foreign genes into the genome plants, but it has some of the disadvantages or some boons as per the electroporation technique. So we has introduced the electroporation techniques. So in electroporation, which is also called as electropermeabilizations, so which is a physical method of introducing genetic materials, proteins, drugs, or other desired molecules, whatever it is, into the cells for various purposes, with the application of electric pulses to create temporary pores in the cell membrane, as she mentioned, as Vanchika mentioned. So it has some electric pulses to create temporary pores in the cell membrane, which delivers the molecule into the cells. So what is the principle behind this electroporation? So the working principle of electroporation involves applying electric pulses to cells, which creates temporary pores in the cell membrane, and these pores allow the entry of molecules such as DNA, proteins, drug, and other substances into the cells. And the application of electric pulses to cells causes disruption of the cell membrane, and the pulses changes in the electrical potential across the membrane, so leading to the formation of temporary pores in the phospholipid bilayer that forms the cell membrane. So these pores allow molecules to pass through the membrane. So once the electric pulse is turned off, the pores start to close and the cell returns to its normal state or position. And some of the molecules taken up by the cells are trapped inside the membrane, the seals. If the electric field allows the membrane defects to reseal, it is reversible electroporation. While it defects persists, it is a irreversible transportation, electroporation, sorry, leading to the cell death. So here are some steps for the electroporations. So first we'll prepare the cells for the electroporations. Then electroporate the cells. After electroporation, we'll recover the cells and culture and harvest of the transfected cells. And we have some advantages of the electroporation above any other techniques, which are electroporation is a versatile method that can be used in various cell types, including bacteria, yeast, plant cells, and mammalian cells also. And also electroporation can used to deliver genetic materials into the cells that are difficult to transfect. Also, electroporation provides consistent and reproducible results. So according to these all the reasons, so electroporation is the best technique to insert the recombinant plasmid with the iron and beta carotene gene producing cells into the cassava plant cells. So the researchers might have used electroporation. Sorry, not might have. He have used electroporation. So according to me, option A is the correct one. So moving forward to the next question, which is the seventh one. So here there are some statements given regarding complementary DNA synthesis. So the statement first states that RNA is zapped to be avoided during RNA extractions as it promotes RNA hydrolysis. And the statement 2 states that DNA's enzymes needs to be added during RNA extractions to prevent DNA contamination. And statement C, third states that phenol and guanidine isothiocyanate are responsible for the separation of RNA from DNA and protein. And the statement 4 states that the chloroform is added to lyse the cells and bring out the contents. So which of the above given statement is correct? Either 1, 2, 3, or 4. So let me ask from someone. Then we'll discuss. Brinda. Brinda.
So,
Hanji. So what is your opinion about this question 7? So which of the statements regarding complementary DNA synthesis is the correct statement? Any guess?
option B.
Any scientific guess?
The option B.
Option B. Why option B? Mm-hmm. Anyone else? Why option B?
So actually, RNA is a very volatile, very hard to handle molecule because of its volatile and reactive nature. So that's why, like some of the DNA also may be present actually inside the RNA. So what needs to happen is for the RNA to function properly, then the contamination by the DNA and it should be removed actually. Whatever DNA is left out while transforming it from DNA to RNA.
Exactly. Correct. So here the option, the statement 2 is the correct one, which states that the DNA's enzymes need to be added during RNA extractions to prevent DNA contamination. So here the cDNA synthesis is given here. So that cDNA synthesis or complementary DNA synthesis is a fundamental molecular biology. Am I audible to all?
Yes, audible.
Okay, just give me 2 minutes. Okay, I'll be back. हाँ जी. So we were on question 7th, cDNA synthesis. So first we'll get to know what the cDNA synthesis is. So cDNA synthesis is a fundamental molecular biology technique used to convert RNA into cDNA. So this process is crucial for various applications, including gene expression studies, cloning, and generation of complementary DNA libraries. So during RNA extraction, so the addition, so coming back to the question, which states that DNA's enzymes needs to be added during RNA extractions. So coming directly to the statement 2, what is the reason behind statement 2? So during what happens is during RNA extraction, the addition of DNA's enzyme is a crucial step to prevent contamination from genomic DNA. So this is particularly important because the presence of genomic DNA is an RNA sample can lead to inaccurate interpretations of gene expressions levels. So when analyzing RNA-based experiments such as gene expression studies or quantitative PCR, which is qPCR, so DNA contamination may rise from genomic DNA released during cell lysis or from incomplete removal of the genomic DNA. So here are some two flow steps are given here. So what is traditional workflow with DNA is one, and workflow with ezDNase enzymes. So according to this, after DNase treatment, the remaining RNA is free from all this genomic DNA contamination, allowing for accurate and reliable downstream analysis. So this approach is particularly critical in experiments where precise quantification of gene expression levels is essential, which ensuring that the obtained RNA reflects the transcriptome of the sample accurately and it is not confounded by the presence of other genomic DNA. So the addition of DNAs during RNA extraction is a standard practice in molecular biology laboratories, ensuring the purity of RNA samples and the reliability of the subsequent analysis. So according to all these principles and theories, so the statement 2 is the correct one, which is DNA's enzymes needs to be added during RNA extractions to prevent DNA contamination. So moving further to the next question, that is 8, is Rohan has purified a recombinant protein. So Rohan wants to check its molecular weight and one of the which of the following methods he could use to check the molecular weight. So the options are SDS page electrophoresis, mass spectrometry, analytical size exclusion chromatography, or all of the above mentioned techniques can be used. So this is an interesting question and very practical question. Anyone would like to answer this? Okay, let me ask someone. Actually, was some issue with my actually there was some issue with my internet, so I was speaking from last 5 minutes. I didn't know. So sorry for the interruption.
Okay, sir, no problem.
So I have explained the entire question 7 in offline mode. So let me repeat that explanation of the question 7th. Let me share my screen. Is my screen is visible to all?
Yes, sir. Visible.
So let me explain the statement second only. So DNA's enzymes needs to be added during RNA extraction to prevent DNA contamination. So let me explain this. So what happened? So during RNA extraction, the addition of DNA's enzyme is a crucial step to prevent contamination from other genomic DNAs. So this is particularly important because the presence of genomic DNA in an RNA sample can lead to inaccurate interpretations of gene expression levels while analyzing RNA-based experiments such as gene expression studies or some qPCR, known as quantitative PCR. And DNA contamination may arise from genomic DNA released during cell lysis or from some incomplete removal of the genomic DNA during the extraction process. And DNase, which is an enzyme that specifically cleaves DNA, is introduced to degrade any residual genomic DNA, which should be present in the RNA sample. So the addition of DNase during RNA extraction is a standard practice in molecular biology laboratories, ensuring the purity of RNA samples and has the reliability of the subsequent analysis. So according to this theories, so statement 2 is the correct one. So not on this, the statement 2 is the correct. So moving forward to the next question, which is 8. So 8 state that Rohan has purified a recombinant protein. So Rohan wants to check its molecular weight. So which of the following methods he could use to check the molecular weight, whether it is SDS-pH electrophoresis, mass spectrometry, analytical size exclusion chromatography, or all of the above. So anyone? Okay, let me ask. Let me ask one by one. Who is Titan? Titan, please answer this question, man. Titan. Okay, no worries. Vaishnavi. Vaishnavi. She is also not responding. Ramanathan. This is a very
Yes,
simple
sir.
question and a very practical question. So if we
Yes,
are
sir.
from some biology background or some life sciences background, can easily answer this question.
Hello, sir.
Hello, Ramanathan. So what is your opinion about this question 8?
Sir,
So
option D.
option what?
Option D, all of the above.
Okay, okay, option D. Anyone else would like to guess?
So option B, mass spectrometry.
Okay, mass
Yes, sir.
spectrometry.
Mass spectrometry.
So it's clearly visible molecular weight and mass spectrometry are both are relatable to each other. That's why you're saying option B. And you guys do not have any idea what SDS page and analytical size exclusion chromatography. So anyone else would like to guess this question?
Sir, I think D should be the right answer.
Okay, D. And kindly explain a bit more. Why option D?
Okay, so in SDS page, we know that the sodium dodecyl sulfate will give the negative charge to
Mm-hmm.
every protein,
Okay.
and the page will decide how the migration of proteins will happen on it.
So,
So
mm-hmm.
and the migration is actually related to the molecular weight, and mass spectrometry we already know it is the mass to charge ratio,
Mm-hmm.
and analytical size exclusion chromatography has beads in it. So it's like the smaller the, like if I give an analogy that if we want to differentiate between a monomer or a dimer, then the monomer uh, uh, it's
Mm-hmm.
actually on the bead size, like which will retain in the bead and which will elute first, and so it is also according to the molecular weight.
Okay, correct. And anything else?
No.
Okay. So let me ask, what is the full form of SDS page? Mm-hmm. Full form. The explanation was
So,
correct. Explanation was damn correct.
sodium dodecyl sulfate,
Uh-huh.
polyagarose gel electrophoresis.
Not it is not polyagrose, it's polyacrylamide gel
Sorry,
electrophoresis.
polyacrylamide. I got confused with agrose gel electrophoresis.
It's
Sorry.
acrylamide gel electrophoresis.
Yeah.
So option D is the right one. So all of the methods could be used to check the molecular weight of the purified recombinant protein. So we'll go one by one. What SDS page, what mass spectrometry, and what analytical size exclusion chromatography are. So we'll start with the SDS page. So SDS page, which stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. It is a type of electrophoresis commonly used to separate proteins according to their molecular weights. So here, as you can see, this KDA is given. This is the molecular weight of any sample A, B, or any recombinant protein, or whatever it is, whatever the sample is. So here, the separate protein according to their molecular weights. And SDS is used to eliminate the influence of a protein's shape and charge as it migrates or moves through the gels. And sodium dodecyl sulfate is a negatively charged detergent that both unfolds protein and coats them in a uniform negatively charge. So in addition, in reducing agents such as beta mercaptoethol is there, called beta mercaptoethol, ethanol, sorry, mercaptoethanol. So which is used to break any disulfide bonds. As you can see, the combined use of sodium dodecyl sulfide and the reducing agent which allows each protein to run through the gel based solely on its molecular weight. So in this way we can use the SDS page to identify the molecular weight of any recombinant protein, which Rohan should have used. Rohan. And the next method which stated was mass spectrometry. So here, as you can see in the diagram, so one m by z is there. So anyone can explain what m by z is? Hmm. What is this unit m by z? It's
Sir,
mass
mass
to
and
charge ratio. Hanji, correct. Mass to charge ratio. So let me just explain what mass spectrometry is. It's a powerful analytical technique used to determine mass to charge ratio of ions, which providing detailed information about the composition and structure of the molecules. And it based on the principle which involves several key steps. The first is the sample is ionized commonly through the techniques like electrospray ionization or matrix assisted laser desorption or ionization, we can say. So in ESI, which is electrospray ionization, the liquid sample is sprayed into a fine mist, and in MALDI, which is matrix assisted laser desorption or ionization, the sample is mixed with the matrix and irradiated with the laser to produce ion. So uh, in this way, the molecular weight of the recombinant protein has been identified by the Rohan. And the last method, which is analytical size exclusion chromatography. So in this method, in layman language we can call this SEC, size exclusion chromatography. So here also in our lab we can call this SEC, not analytical size exclusion chromatography. Only we say SEC. So SEC is an gel filtration chromatography, which is a powerful analytical technique used in biochemistry, molecular biology to separate and characterize macromolecules based on their particular size. And the principle behind SEC relies on the differential exclusion of molecules from some pores in a porous gel matrix. So here, as you can see in B1, so kilodalton, some units are given here of some particular protein expressions or some whatever the bands shown here. So it is differentiating according to their molecular weight. So in this way, Rohan might have identified the molecular weight of the purified recombinant protein, whatever he has isolated. So based on this theories and principles, so the option D, which is all of the above mentioned techniques can be used by Rohan to check the molecular weight of the recombinant protein. So option D is the right one. And moving forward to the next question, that is 9. So here Suresh has purified N-terminal his tagged recombinant protein by affinity chromatography. So however, the SDS page showed bands in additional to the target protein. So the question is, how might Suresh can improve this purification step? By including protease inhibitor during purification, or by decrease the volume of washing, or either by use chelating metals in the column, or decrease saline concentration in the binding buffers. So anyone would like to involve in this discussion? Okay, let me ask someone. Vanchika Garg,
Yes, sir.
what is your opinion on this?
Sir, I'm still thinking about it. If you just give me a minute.
Take your time, take your time, take your time. Anyone else?
So maybe A.
And what is the reason behind option A?
So not having much of an idea actually.
Hmm. Go ahead, go ahead.
So just like it just felt ki like protein is there, so protease will somehow like regulate the size or whatever is coming on that
Hmm.
plants.
Okay, exactly. The option is the right one. So, who was thinking? Vanchika,
Yes, sir.
now done with your thoughts?
Sir, I think he is right about it.
He is right. So, Suresh can improve his purification step by including protease inhibitors during purifications. Okay, so let me explain this and why he should have included protease inhibitor during purification step. So, if Suresh has observed some additional bands on SGS page besides the target protein after purifying N-terminally his tagged recombinant protein by using affinity chromatography. So there are several strategies he can employ to enhance the purification step, and he can obtain a more pure sample of the desired protein. So first he should have optimized the washing conditions during the affinity chromatography step. Then after step 1 he should have increased this stringency of wash buffers by adjusting the factors such as chromatography, salt concentration, pH, or the type of detergents can help remove non-significantly bound contaminations. So as you can see in the given figure, some general purification of histidine tagged, which is his tag proteins are given here. So let me explain how does histidine tag protein purification works. So histidine tag proteins are commonly purified using immobilized metal affinity chromatography, which is based on the interaction between amino acid residues and divalent metal ions immobilized on resins actually. So histidine interacts most strongly, and histidine tagged protein have extra high affinity because of the multiple histidine residues, and the chemical imidazole is used to elute histidine tagged proteins. A lower concentration can also be used to wash resins prior to the elutions. And the next question comes is, when should Suresh has used histidine tagged protein purifications? So the main question is protease step. So the histidine tagged protein purification can be used as the only purification step for applications that do not require proteins with extremely high purity. So when the highest purity is needed, so this protease inhibitors can, sorry, what is protease, not inhibitors? Sorry, protease inhibitors should have used at the first step in the multi-step purification. So it is also suitable for purifying dual tagged proteins while with a histidine tag at one end at a different tag at the other end. So by using these some purification steps, so Suresh must have histidine tag protein purification must be used by Suresh. So according to all these theories and principles, so he should have go on with the option A, which includes the protease inhibitor during purification step. So am I clear?
Yes, sir, clear.
So moving further to the next question, which is the last question, question 10. So here, Nikita has extracted RNA from certain yeast cells. So Nikita wants to quantify it for further downstream experiments. So she dissolved the extracted RNA pellet in 100 microliter of distilled water. So further, she diluted the sample in distilled water. In the ratio of 1 is to 50, the absorbance of at 8 to 60 came to be 0.46. So what is the Nikita's total yield of RNA in microgram? Anyone would like to answer? This is a simple calculation. Just a simple calculation. Siddharth.
Yes, sir, just looking for it.
Mhm. So meanwhile, anyone? Anyone can answer.
So 92 microgram.
Okay. And how 92 microgram? So what is your strategy to
So there'll be
calculate
one.
this?
So there'll be one formula mostly A to 60 into 40 microliters into 100 is equal to the ratio.
Exactly. So is my screen is visible?
Yes, sir, visible.
So I have calculated this. So just tell me, is it correct or not?
Yes, sir, it's correct.
So the answer is 0.92, sorry, 92 microgram. So do I have to explain this? Or actually, this is a simple calculation. So okay, so let me explain. So since the absorbance reading at 8260, so which is 0.46, so it is possible to determine the concentration of RNA in the original unlead diluted samples. So earlier the formula concentration is equal to absorbance into 40 microgram per ml. So after calculating its concentration came as 18.4 microgram per ml. And the next, it is essential to account for the dilution factor. The specimen underwent a dilution of 1 is to 50 as said by the as wait as said by Nikita. So resulting in a dilution factor of 50. So here the formula for the concentration in original sample is concentration in diluted sample multiplied by dilution factor. So according to this calculation, it came 920 microgram per ml. And to determine the overall RNA yield in micrograms, one must multiply the concentration by the volume of the initial sample. So the total yields formula is concentration multiplied by volume of the original sample. So which came 92 microgram is the total yield. So the according to this calculation, so the option C is the correct one. So the 92 is the total yield uh, by the Nikita. So am I clear?
Yes, sir, clear.
Okay, so I'm done for the day. So there's one announcement. So the session which we are going to have on 27th. Okay. 27th session is postponed to the next session, which, am I audible to all? So this is an
Yes,
information.
sir, audible.
So 27th session is postponed to the next week, which will be held on the 5th March. So on 5th March, I'll be taking both the sessions of 27th Feb as well, also with of 5th March as well. Okay. So on 5th March, we'll go through the two assignments one by one.
Okay, sir.
Uh, so if you have any some friends on this with the same session, just do let anyone know. Okay. Someone will ask. On 27th Jan, I'll not joining the session. Okay, am I clear?
Yes, sir.
Okay, so let's conclude this session now. So thanks to all the students. So you can leave one by one. Okay, thank you.